And then 2 reading responses for another class. One of them was already late, so I gotta get that one. The what good cat? The what cat? No.
Aha.
I did wait back.
All right. Are we good on time? All right, let's go ahead and get started. Today we're going to be talking about diversity and how we quantify it, how we compare it. Hopefully that prep homework was useful for you. We're going to go over a lot of the same concepts today, but of course we should start with our prairie organisms of the day. Just to let everyone know who's watching this later, I happen to leave my iPad at home today, so I'm recording on the laptop. I'm going to try to speak up, not just for the people in the back, but so if you watch this later to review, you can hear me on Zoom. So that's what I'll be going for. So our prairie organisms of the day, let's start with the grass. So far, I think all the grasses we've shown you have been what we call C4 grasses. Grasses that typically do better in drier or more water-stressed environments. This is a C3 grass, Caleria pyramidata. Caleria grasses are known for their flowers. So you might not always think about grasses having flowers, but of course they are flowering plants, but they're pollinated by wind. So they don't need to have showy flowers to attract animals to disperse their pollen. But in prairie June grass, these inflorescences, these spikes that have the flowers, can be quite showy, kind of a bright sort of a pale yellowish color that is pretty attractive. And it's a clump forming grass. So you've got an underground stem with lots of branches that are coming up, bearing leaves, and they make that kind of a clumpy look. C3 grasses tend to do better, as I said, in cooler and wetter environments. They tend to do well early in the season. And the further north you go, the better C3 grasses tend to do because they tend to have a cooler growing season. They tend to flower June to September. And this one is maybe a little more unusual for a typical C3 grass in that it's reasonably drought tolerant, which is part of why it does well in Kanza along with all of those C4 grasses. So that's prairie June grass. And if you just look up June grass inflorescence or something like that, you can see kind of the complicated but elegant looking flowers of prairie June grass. Of course, we got to do a prairie forb as well. Did I show any of you this thing, Salvia, when we were doing in the field at the beginning of the semester? These have very interesting stamens. So salvias are closely related to mints. So anything you ever eaten with mint is probably in the Lamiaceae, and salvia is also in the Lamiaceae. That's the mint family. And salvias have really interesting stamens, which are the pollen-producing organs. When an insect approaches a salvia, it kind of pushes a little lever that's on the stamen, and that pushing that little lever makes the little pollen-producing structure, the anther, come down and bop the insect on the back or bop it on the head. And then when it visits another flower, the pollen receptive organ, the stigma, just has to touch it in that one spot. So it can be very precise in terms of where it places the pollen. You don't have to dust the whole organism with all the pollen you can. If you place it perfectly, then the next flower visits gets it right off that same spot. So salvias are quite fun in that way. And if you open up one of these flowers, you can manipulate that little lever and see the stamens come down. So they're kind of fun. Did any of us do that this year in the field? Usually I find a group and I show one of you guys. They flower July to October. That's why we saw them at the very beginning of the semester out there. They form these really nice blue blooms. As with most members of the mint family, the leaves are aromatic. So one of the things you can do to get to know plants, to get your ID skills up, is to not just get to know the flowers and the leaves, but take the leaves and crunch them up and smell them. in some families, especially the mid family, you'll get to know those scents. They help with your memory of knowing what plant you're working at. They can be kind of tall, and the roots can also go quite deep. So having deep roots is a good adaptation for water stress growing in a prairie like Alfonso. So and they're pollinated by bees, bumblebees, as well as butterflies.
Is there something about plants that are small?
Oh yeah, so I probably would have talked to you guys about that. Most members of the mint family, you can recognize not just by their flowers or the scent of their leaves. If you feel the stems with your fingers, they're square, or they have 4 sides. That's because the leaves come off in pairs, and each pair is 90 degrees offset from the other. So that's another good tip for recognizing this family, especially. Do I have anything else here? Yeah, good. All right, and our prairie animal of the day is the western meadowlark. So we're in Kanza, we're kind of at the eastern edge of its distribution. It does well in grasslands and drier environments, but all the way over to the forests at the west coast as well. Attractive bird, Sternella Neglecta. This is in the blackbird family, so it's the same family as blackbirds. They tend to feed on the ground in grasslands and pastures and around like wetland edges. Oh yeah, so this was named, the scientific name, Sternella Neglecta, was given by Audubon. So have you guys heard of the Audubon Society? It's a famous society devoted to the protection of wildlife, especially birds. John James Audubon was a big advocate of birds and described several species, including this one. And he claimed that most explorers and settlers who had ventured west of the Mississippi after Lewis and Clark had overlooked this common bird. So that's where the neglecta comes from. Many species, people are getting to know all the organisms, but no one talked about this one. He thought it was important people know about it, so called it Sternella Neglecta. Like other members of the blackbird family, meadowlarks use a feeding behavior called gaping, which relies on strong muscles that open the bill. So they'll insert their bills into a tree bark where there might be some insects, or into the ground where they might find some grubs, and they'll open their mouth. And by opening their jaws, they create that space where they can access the food item. So they have unusual musculature in their face for opening their bills. And so the behavior is called gaping. You can see that with with the rather large, rather straight bill. There's a lot of force there to get into some bark or something like that, into the soil. And that allows them to access insects and things that other organisms can't access. Have you guys heard about birds adapting their beak structure to better access resources? No. Excellent. All right, so those are organisms of the day. Any questions about these? The meadowlark, the salvia, or the prairie June grass? If you look ahead on the schedule, you'll see that photosynthesis is coming up, and that's where we'll really dive into the difference between C3 and C4 photosynthesis. So now I'm going to try to see if I can switch slides here. So measuring diversity. That's what today's all about. We're going to define some terms, which you all have already done in your prep homework, which I'll collect at the end. We're going to talk about the species-area relationship and the effect of sampling on the ability to estimate species diversity. We're going to talk about some of the different ways that ecologists measure diversity or try to estimate diversity and how they compare it. We're going to think about creating and interpreting figures that show rank abundance of different species. So you'll learn what abundance is, you'll learn what we mean by rank, and you'll learn how to read the plots that are in the V-Net All paper. And then finally, we're going to talk about the effects of bison on species richness at Kanza Prairie and the reason for these effects. So what do we mean by biological diversity? Well, what I want to say about this is a little more than what's on the slide here. The way we use biological diversity is kind of context dependent. So if you look up, say, the UN's definition, you'll get something pretty close to this. The total variation of all organisms, right? So the genetic, phenotypic, ecological, or maybe community is better here, community variation of all living organisms. So a really broad topic, just any biological variation, almost any biological variation can fall under this idea of biological diversity. But in ecology, and in the context of this study in particular, we're going to be talking mostly about the number of species in a community and the abundance of those species in the community. Abundance, of course, may be measured in number of individuals or the total biomass or cover of plants. There's lots of ways you can get at it, but which ones are more common or which ones are there more of and which ones are there fewer of? That's what we mean by abundance. And then the other element is exactly which species are there. So you might have, for example, 2 classrooms that have the same number of students and the same number of guys and girls in the classroom. But the students themselves are different, so the classrooms are different. So you can compare the relative abundance of guys and girls. You can compare the number of students in the classroom, but also you can just look at how many students are in both classes, right? That's what we mean by composition. So richness, abundance, and composition, all those go together to capture this concept of biological diversity in an ecological context and at cons of prairie. So what's a community? Actually, I was going to ask you guys before I did that. What's a community in ecology? I think I attempted to ask this last time I lectured. But what do you guys think? What makes a biological community? How is it different from other things that we've talked about so far? Species interacting with each other. Yeah, so it's different species interacting with each other, which of course I showed you a second ago. It's in a defined area is the other element of that. So when we think about a community, there's some boundary. And as a researcher, you might impose that boundary, or there might be a really obvious boundary in nature. So for example, the community of organisms living on Isabella Island in the Galapagos. So there's a really obvious boundary there. Maybe not the shore, maybe some animals go out and swim to get some food, but the shallows around the island is a pretty good boundary for almost all the organisms in that community. What are the boundaries at cons of prairie? Yeah, so at one scale, the boundaries are the fence lines or the fire breaks around the watersheds. So you can also think about the boundaries of a community being operational, something that you have to impose as the researcher in order to make your questions answerable. Some organisms go further over the course of their lives. Some organisms stay in one spot. So even on Isabella Island, right, you're going to have seagulls in the community. They might go from one island to another, and so they don't really care where the shoreline is. But all the bumblebees that pollinate the yellow cotton plants are going to stay on the island. So you can get fuzzy boundaries. You should know the relationship between a population and a community, which is maybe obvious in the definition here. And Caden said that the organisms are interacting. What are some of these interactions might be direct or indirect? Anyone think of a direct interaction among individuals of 2 species? Any example of direct interaction? Yeah, get us started.
Predation.
Predation, right? One species actively eating the other. That's a direct interaction. Can you guys think of any indirect interactions where the activity or population of one species affects another one, but not because they directly interact, but through some other intermediate? Yeah.
And thinking of the realm of plants, competition for like nutrients in the soil or water.
So if you've got 2 organisms that are going after the same shared pool of resources, and they're accessing that same shared pool of resources, we still call that a direct interaction. You guys think of an indirect interaction?
Sunlight, like the honeysuckle trees and that.
Again, if you're interested in multiple species in the same area going after the same resource, in this case light instead of soil nutrients, we still call that direct. How might a predator, to build on the predation example, affect the population of a plant? Okay, so decomposing animals might add nutrients back into the soil and contribute to that resource pool that the plant is going after. That's a pretty good one. That's not what I was thinking of, but that's a reasonable one. And then seed. So how would seed dispersal work in this situation?
Well, you know,
Yeah.
the animal eats it and then does its business. Plants.
Again, that's a direct interaction. So the animal is directly getting the baby plants, putting them where they need to go to grow. Yeah.
I'm gonna receive animals that eat the plants.
Yeah. If the predators eat the animals eat the plants, what's the net effect of more predators on plant growth? Yeah, it would go up, right? So the more predators eat the herbivores, the fewer herbivores that are eating the plants, and the more the plants can grow. So if there was a predator eating your seed disperser, Lily, then you have the indirect interaction between the predator and the seeds. So we can have both indirect and direct interactions. And that one we've just talked about is called a trophic cascade. So trophic levels are levels in a community where all the organisms use the same resources. So you've got the plants as the bottom trophic level, and there's herbivores, and there's predators. There might even be predators of predators. Those are what we call trophic levels. And a trophic cascade is where you can jump across one trophic level to another based on the interactions of the species at each level. Predator, herbivore, plant. There are other kinds of indirect interactions as well. Yeah.
What about the seeds that like stick to us? Little, like how you were out in the prairie, your pants were like covered in seeds. What would that be?
That's also a direct interaction, but if the seeds don't affect you, what kind of interaction is it? If the seeds don't affect the animal, but they benefit the plant because it's being dispersed, what is it? One more time. I can't hear that. Mutualism is when they both benefit. But if you aren't bothered by I know many of you were, but if you aren't bothered by the little sticky seeds on your pants, what kind of relationship is it where one benefits and the other one's unaffected? You guys remember that one? Commensalism. Yeah. So it's still a direct interaction, but not a big cost to a bison that has a few, grass seeds stuck to its coat. I already talked about this. The boundaries of a community can be gradual. They can be more important or less important for different species in the community. So often, ecologists have to define their own boundaries for a community. So to help you guys put this into a broader framework, I've put in the classic hierarchy here that's in an ecology textbook. You'll find it in your textbook if you look for it. Organisms comprise populations, right? A group of populations in the same area potentially interacting makes a community. And then communities, if you add in not just the species that are interacting, but also the attributes of the physical environment, the climate, the nutrient flow, like leaching, we talked about that, incoming sunlight, the amount of energy, all those kinds of things, the cycling of nutrients through the organisms as they decompose and provide nutrients back to the plants. All those abiotic factors plus the community gives you the ecosystem. And we'll talk about nutrient cycling later on in that ecosystem framework. And then, of course, you could have ecosystems, you could lump those together into biomes. And we already talked about how the biomes are defined basically by climatic zones on Earth. There's the tropical zone, which includes rainforests and seasonal tropical forests. There's the desert biome. There's temperate forests and prairies in the mid-latitudes, boreal forests and Arctic environments at higher latitudes. So those are your biomes that can have many different ecosystems in them. So here's our Kanza community. What kinds of organisms do you see in this image? Bison, right? Our animal eyes go right for the animals. Yeah, all kinds of different plants. Look, we got some with the yellows here. We got this might be some dogwoods, maybe. We've got all different kinds of species of grasses. You can almost detect some blues. There's something else up here. What do you notice about the distribution of species in this image? The spatial distribution of species. It's patchy. That's typical for almost any landscape. And you'll see organisms have a patchy distribution. So keep that in mind as we start talking about heterogeneity. What kinds of interactions do you think are present in this image?
like they were using the grass around it.
Yeah, so bison are going to be an herbivore on the grasses. What's another interaction you're going to see there? Competition among the plants, for sure. There might be different combinations of resources in kind of these low depression areas versus along the fence line versus up on the hill slopes here versus down in the valleys where the water's flowing. In fact, you can see where the valleys are coming in, you get more of these larger plants, and then up on the hill slopes, it's more grassy. What else? How else are the bison going to interact with the plants? We've got bison consuming them, we've got plants competing, and what's the other way that bison influence the plants? Fertilize it, and what do you say? Yeah, they're going to die, right? So they're going to defecate and urinate. They're also going to die. And what else are they going to do when they play and get those parasites off their coat? I don't know what that behavior. Yeah, the wallows, right? So there's lots of ways they're going to interact with the environment. Okay, so now that we have a good idea of a community and how you can just look out there and start thinking about what components there are, what kinds of interactions are taking place in the community, let's give you guys one more set of vocabulary. So community refers to a set of locally interacting species. But what you'll find is that a lot of times people will talk about the plant community or the bird community or the mammal community. And that's fine, that's the language we use. Another way to refer to something like that is an assemblage. So let's look at the three rings here and then think about the intersections. So a clade is something we haven't talked about a whole lot yet, but a clade represents a group of organisms, all of which are descended from a common ancestor. So for example, all dogs in the genus Canis evolved from a common ancestor, also in the genus Canis. So if you find a common ancestor and circumscribe a group that includes all the descendants of that common ancestor, that's a clade. Another word for a clade is a taxon. Ideally, all taxa are clades. It's not always the case, but that's our goal as systematists. And you can have clades at different ranks. So you could say all vertebrates are a clade. That's all animals with backbones. Or you could say all dogs are a clade. You could say all flowering plants or all grasses. So they can be nested sets. Clades can be nested sets of organisms. And then the last one here is a guild. So a guild is a group of species that exploit similar resources. So you could say all of the rodents in an environment that eat seeds, eat grains. So you can be more specific than a trophic group. So plants, right? They're all taking in energy from the sun, taking in carbon dioxide from the atmosphere. That's a trophic level. All plants are a guild. But you could be more specific than that and say just the trees, right? So that's they're all plants. They're still primary producers, but they're interacting with the physical environment in a specific way. They're being trees. They're competing for light. They're growing tall. So that could be a guild. Just like herbivores, you could say all herbivores are all seed predators, or you could say all the rodents that eat seeds. That could be a guild. And then, of course, you can be specific. So maybe you want to say, well, I'm only interested in all the plants in this area. So that's an assemblage. Or you say only the plants in this area, and I'm going to acknowledge that they all are doing photosynthesis. So then you have an ensemble. That's just the plants in your area. This is probably the term that's used the least, but I want you guys to know community, clade, and guild. Those are the important ones. And know that you can sometimes hear someone talk about a community, but they're only talking about one group of organisms, like the bird community. So technically, that would be an assemblage. Bird community is fine. Questions about these three ways of thinking about how to group organisms? Seem okay? All right, good. All right, so let's go to the ways of measuring and comparing species diversity that are relevant for the Veen et al. Paper. The first one is species richness. Someone tell me what species richness is. Someone, yeah.
Like number of species.
Yeah, the number of species in a certain area. It's a pretty simple one, right? It's easy to interpret. If you want to communicate with someone who's never had a biology class, as long as they have some vague notion of what a species is, they can understand what species richness is. It's centrally important for conservation biologists and also for policymakers who they communicate with to make policies that can protect species. But it's also a little bit limited. It's only one aspect of diversity. Remember, we talked about diversity also includes composition and abundance. Both of those things are neglected if you just count species in an area. But let's think about it for a moment. Let's say that you need to measure species richness in an area because you've got to communicate that information to a policymaker. Well, to measure species richness, we need a sampling strategy. And one of the common ways is to use quadrats. So a quadrat is just going to be our sampling strategy where this is our community, and we're going to set up these quadrats to sample individuals, identify the species those individuals belong to, and then assess the richness or estimate the richness. There's a few other ways we can do it. We can use the line intercept method or the point method, but this is the quadrat method. So you put out a quadrat, you count the individuals, identify their species. Then you throw another one and another one, do the same thing, and notice they're randomly distributed. You want to get a fair sample of the species that are present in this whole community. So they appeared in kind of a random, or maybe a stratified or haphazard way. And once you know how many species there are, you can, in those, you can then have some estimate of what the species richness is for the whole community. If you've got species that are distributed across this whole area, and you sample some of them here, and then you go to your second spot, do you think you're going to get all new species, or do you think you're going to get some repeats? Yeah, probably get some repeats, right? Because you're specifically picked a community that's somewhat uniform. And so the species that are present in that community should be distributed maybe evenly, maybe unevenly across the whole community. So you should get some repeats. And here's an example of a way you could look at the relationship between the total area sampled and the species richness that you measure at a place like Kanza. So you could have a whole bunch of plots, 1, 2, 3, 4, 5, 6, all the way across here. Each one of these plots has a certain area. So each time you sample another one, or by the time you sample the second one, you've doubled your area that you've sampled so far. And eventually, as you get to number 30 here, you've probably sampled about a third of the total area on the map in this, on this one little ridge here. So what do you expect the relationship is going to be between number of species you've detected and the total area that you've sampled as you go from 1 to 30? Any guesses as to what that's going to look like? Say that a little louder. Well, remember we're talking about communities, so species. A third of the, so Hayden estimated that you would get to a third of the total richness of the area. After you've sampled 30 of these, do you think that if you go right here, you're going to, how likely is it you're going to find a species that you hadn't ever seen before? Unlikely. So if that's the case, do you think a third of the species is a good estimate of how many you've seen by the time you get to 30? Probably catch most of them by that point, right? So each additional one you add on, you're not actually gaining a whole lot of new information. But what do you think the graph would look like? If you graphed kind of number of plots or total area of all the plots you've looked at and the number of species you've detected, what shape might that take? What shape might that? Did you look ahead? A little bit. So Caden said a curve with a plateau. And that's exactly what we find. Initially, each new sample you take, you're going to get new species. And there's going to be some slope to that line. But eventually, once you've detected almost all the species that are there, adding new samples or adding new quadrats isn't going to gain you more species. You'll still get new individuals you never saw before, but they're going to belong to the same species that you already sampled. So that's why you start to get this curve. What do you think the asymptote is going to be here? Or how would you interpret the plateau? That's what I should say. How would you interpret the plateau on this graph?
The same thing.
Say it again.
You're finding the same thing. You're not finding.
Oh, yeah. Okay. So Kane said you're finding the same things for those of you on Zoom. He said the plateau means you're finding the same species as you keep sampling. Yep. If you could estimate where this line becomes perfectly flat, what will you have measured at that point? The total number of species in the area, right? So that's one way we can interpret that. That's your total. How do you think, well, let me, yeah, okay. How do you think spatial heterogeneity on the landscape, or heterogeneity of resources, let's say, might change the nature of this relationship? Or let me ask it this way. Okay, so what I've done is I've just traced the curve on the board, and I'm going to ask you all to think for a moment. If you had a different community which had more spatial heterogeneity of resources than the one represented by this curve, how might that curve look different? I realize this is a challenging one because I'm asking you to imagine a community and then imagine another community that's different. And the difference is in heterogeneity, which is kind of a new concept so far. But if you've got more variation in the availability of different resources in a given area, how might you expect this curve and the two attributes of this curve to change? Well, you're always going to eventually get to the point where you sample everybody in the area. So there will be a plateau, but how might it be different? Clara, do you want to add to it? Yeah, so one thing is if the environment is more heterogeneous, Clara said that maybe it's going to be steeper in the beginning. So as you go from this spot to this spot, the availability of resources is more different in a more heterogeneous environment. And so which species can do well in those different conditions are going to be very different. So you'll accumulate different species more rapidly initially. Might anything else be different? Yeah, so good point. So if there's less heterogeneity, then you might expect that initial slope to be more shallow if it's a more even community. Yeah. Okay, so Claire went to another thing that I was going to talk about in just a moment, which is where if you sampled a lot and then you kind of ignore the order in which you sample to create this curve, you're going to get a smooth curve. But if you plot how many new species you found with each sample in order, so sample 1, sample 2, sample 3, sample 4, that's called a species accumulation curve. And in a species accumulation curve, it looks like a stair step. And the height of the steps is going to be related to how heterogeneous your environment is. So Claire kind of got ahead there. I think actually I might have taken that one out. But if you see the stair step, you're looking at a species accumulation curve or a sampling curve. Whereas here, we're ignoring the order in which these appear and kind of saying, well, if you did it 1,000 times, what would the curve look like overall? So what King was getting at earlier was that maybe something about the total number of species could change. And potentially, with more heterogeneity, you could have more combinations of resources, which creates more possible niches for species, and you could pack more species into the environment. So it's possible that you could also have a higher plateau in a more heterogeneous environment. And that's how it's typically presented in textbooks, and that seems to be generally true. But spatial heterogeneity in habitat conditions or resource availability usually promotes species richness by increasing the opportunities for other and different species to do well in that environment. But eventually, as heterogeneity keeps increasing, the kind of amount of habitat space in a community for each species goes down. So eventually you might get to a point where with really high heterogeneity, species diversity starts to decline. But generally, the typical rule is with more heterogeneity, you get a steeper slope, and maybe you also get a higher asymptote. If you have, just to clarify that last point about species richness going down with high heterogeneity, imagine if you have a really heterogeneous environment and you have all these different little subhabitats within that, or different combinations of resources available. Eventually, the species that are occupying all those different habitat patches are going to have smaller and smaller population sizes because there's more different kinds of habitat combinations. And if your population is really small, the odds that you stochastically or randomly go extinct just by chance goes up. If there's only 2 of you, any random thing could knock out both of you and then the species is extinct. But if everyone's population is in the hundreds, then you're probably not going to go extinct randomly. So that's why there's that upper limit to this pattern. But generally, that's the change in the asymptote and the change in the steepness of the curve. And so then this question about are species evenly distributed on the landscape? This speaks to Clara's insight, which was that if you put your number of species in order, or number of samples in order, then that clumpy distribution will mean that you'll get a stair-step condition. So maybe you're in a patch of big blue stem, and then your next plot is in the same patch of big blue stem. And then your third patch is in the same patch of big blue stem. But then suddenly you're in a patch with dogwood. So you're going to get very different species because of that patchy distribution that you saw in the picture early on. There's another place where we saw the relationship between number of species and total area in this course. Does anyone remember where we saw a relationship between area of a habitat and the number of species in that habitat? It was an area that we didn't do too well on the first exam. And I think that was, I don't know, it was one of my topics, so that's why I want to revisit it here. I want to make sure you guys get it. Where did we see the greater the area, the more species could be present? So Katie said in the Galapagos, it was generally that island biogeography stuff where we had the graph with the 2 lines that curve like this. Yeah, so that came from this relationship between area and number of species. And there's a few quotes here that I won't read you right now, but be sure you go back and think about what they're each saying. This is one of the important ones here. As the area increases, the more types of environments are likely to be encountered. So what might be a homogeneous habitat on one scale of meters, if you zoom out so you're seeing kilometers, there might be patchiness there. So as the area increases, the more you can detect heterogeneity and the more types of environments you're likely to sample or encounter. And because species are not uniformly distributed across those environments, the number of species that you see will increase with area. And we can see, here's a couple examples of that. These are the West Indies. So we got the Greater Antilles, the Lesser Antilles, and the Bahamas, or what's the name of the archipelago? There's an archipelago here that includes the Bahamas and a few other islands. And together, these are all the West Indies. And we can look at an assemblage, basically the herps, lizards, snakes, frogs, all the reptiles and amphibians. And we can see the relationship between the number of species on an island and the area of that island. And the nature of this relationship is basically the larger the area, the more species. But eventually it asymptotes because there's only so many species you can have on a given island. So the bigger the island, the more species. And if we transform this graph so that we're going up by orders of magnitude instead of incrementally by 1 extra kilometer squared, but by 1 kilometer squared, to 10 kilometers, to 100 kilometers, to 1,000 square kilometers. Here's Cuba. Notice how close Cuba and Hispaniola are on here. But on the map, that's Cuba and that's Hispaniola. Cuba is actually quite a bit larger. And here's Hispaniola and Puerto Rico. Of course, Hispaniola is a lot larger than Puerto Rico, but look how they show up on this log scale. They look closer together than you might think if you put this on a normal scale. So the point is here, you can see that as area goes up, the number of species goes up. We saw that on the Galapagos as well. So here's all the vascular plants on the islands of the Galapagos. A plot from Preston, 1962. Here's your different islands. There's Isabella, the big one, and it has the most species. Floriana, which is right next to Isabella and is also rather large here, has a similar number of species because they can move between the 2 islands. And then the small islands like Pinta down here. There's Pinta, and Floriana is, where's Floriana? Oh, Fernandina, Fernandina. So you can see that as the total area of the island goes up, the number of species on the island goes up as well. And here's some of our Galapagos plants, the cotton, right? All those yellow things pollinated by the bumblebees. All right. So now that we've thought about species richness, let's think about some other ways that we can compare communities, not just number of species, but other ways that also capture their abundance. So we've got relative abundance and species evenness. You'll see for relative abundance, we're going to use this P sub I. P means proportion. So what proportion of individuals in a community belong to one species? And the little I, remember how we've been talking about the little subscript I? What does that usually mean? Initial is one use, but when you have to like do an equation where you sum across a lot of numbers, what does it mean in that context? Yeah, index. So what P sub I means is it's the proportion for each species in a community. So you have to index through all the species in the community. It might be 11 species or 200 species. And then evenness is just a measure of how equally the individuals are distributed across the population. Do all the species have the same number of individuals, or some species have a lot of individuals, other species only have a few individuals? That's what evenness gets at. It's very similar to abundance. It's just another way of thinking about it. So let's do it visually instead of all this text. Imagine these are our 2 simple communities. Which community looks like it has higher plant species richness? Why are they the same? Or yeah, I guess that's the easy question, right? Because if you recognize the different species based on their morphology, we see 1, 2, 3, 4. Okay. 4, 3, 2, 1. Right? It's the same. 4 species in both. Community A and community B. But which one has higher evenness? Which one would score higher on the evenness board? Yeah, A, right? So what you're seeing there is just that here, each species makes up a quarter of the community, 3 out of 12 plants. Whereas here, 2/3 of all the plants in this community belong to one species. So it's less even, and this one's more even. Good. So now if you want to think about diversity, it has both the abundance information and the richness information. Which community is more diverse? Yeah, the one that has higher evenness. This is why it's important to have a measure of abundance in your assessment of communities, and why just using species richness can be limiting when you're trying to decide which community should I protect. They both have the same richness. Which one's more diverse? Here, this one random thing could happen, and suddenly this species goes extinct. If one random individual of this species dies here, the species persists. So let's think about how we do this in terms of organizing data. Here we have 2 communities, kind of like the pictures we just had. and we've got lists of species. So just based on what's here, which community seems like it would have higher diversity?
One.
Yeah, one with more species, right? But if I add in the number of individuals of each species detected in both of these, and then I figure out what proportion of those individuals are in each species by figuring out the total here, and then dividing that by that to get that. Now, which one, in which community would you be more likely to encounter new species with each additional sample? Yeah, community 2 is more even. Here, your first site was probably going to have mostly just big blue stem and Schizocarium. And your next site is probably also going to have those two. And then every once in a while, you get a site that also has one of these. And eventually you get to all of them. Whereas here, you'll get a few of each. Your next site, you'll get a different few of each. And then your next site, you'll get another few of each. So you're more likely to encounter new species as you sample initially over here. So which one seems more diverse now? Someone says 2. Anyone agree with that or disagree with that? It's a challenging question. So I take your silence as reflecting that it's a hard thing to answer, right? How are you going to mentally integrate both the number of species and the evenness just by looking at a column of numbers? Maybe this one is more diverse, but how much do you value the addition of each new species, even though this one has higher evenness? Well, one way we can do that is to actually make a plot. This is called a rank abundance diagram. And when you scale the y-axis here to proportion instead of number of individuals, but to the proportions, we call it a Whittaker plot, named after an ecologist, Robert Whittaker. But really, it's just a form of a rank abundance diagram. And the species rank, anyone guess what we mean by species rank based on this graph? What order do the species appear in on the x-axis? So that is the order, but how do you know which species is number 1? The most abundant. So they're ranked in order of abundance. That's what we mean by a rank abundance distribution. By looking at this graph, again, what 2 aspects of a community can you quickly assess and compare? Evenness, right? Which one is more even? Yeah, 2, right? There's not a big change in slope here. Whereas community 1, you got this really steep slope initially. So it's highly dominated by this first species. What's the other thing you can compare besides the slope or evenness? Where did the 2 curves stop? Well, how many species are in community 1? How many are in community 2? So you can, just by looking at the x-axis, you can see species richness. So you get those both, and it's maybe a little easier to compare, and you might say, well, based on this is really dominated by species 1. So maybe I would say community 2 is more diverse. But still, we haven't quantified that, which we'll do in a moment. So think about how environmental heterogeneity might affect species richness or species evenness. We've already talked about richness a little bit. How might you expect a more or a less heterogeneous environment to look in terms of being highly dominated by one species or evenly dominated by many species? Would you expect a less heterogeneous environment, a more homogeneous resource distribution, to be dominated by one species or by many species? If resource availability is the same across the whole environment, would you expect a lot of species or just one species? Why do you say a lot? I want to hear a little bit explanation. Say it again.
So,
Did you say because they're plants?
I said, there's just a lot.
Oh, okay. Because there's a lot of resources.
Uh-huh.
Okay, so think about populations. How does having more resources affect population growth? What does it change? It changes the availability of resources, which are then turned into organisms, right?
Yeah.
So an environment with more resources has what for a population? A higher. Remember the logistic curve? Yeah.
Well, I was gonna say, like, the thing that Paul was showing us yesterday, like, the more excess of resources that can have, the more likely they're able to coexist.
Aha. Okay. So we're going to break that apart. So having more resources, remember, increases your carrying capacity or the capacity of your population to grow. So if that resource supply point is way out here in Paul's diagrams yesterday, the populations are growing, which you would see as having the steep part of that population growth curve from early on. So having a lot of resources just increases the population size of an individual species. But remember, each species needs its own unique combination of resources to grow. That intersection point on the diagrams yesterday, that's unique to each species. It has its own necessary combination, it's optimal, its own resource ratio that's optimal for growth. So if you've got an environment with uniform availability of resources, the species that's best adapted to that condition is going to take over and win. And so you're going to get a community that's highly dominated by one kind of species. But if your habitat is very patchy with different combinations of resources all over, then here one species wins, here another species wins, here a third species wins. And so over the space of the whole room, for example, you would get a more even distribution. So environmental heterogeneity is probably going to increase evenness at a large scale, but then locally in each patch, you'll see highly dominated areas. So it kind of depends on how big your quadrat is, right? Relative to the scale of heterogeneity in the environment. And that's one of the challenges as a researcher is to match your sampling strategy to the environment that you're working in. How do you think disturbances might influence species richness or species evenness? There's a couple ways you could answer this, so don't be afraid to be wrong. Someone other than Sonia to get us started. Yeah.
Well, there's some sort of like,
Yes. Okay, good. So a disturbance is going to remove individuals and probably some species, so your richness is going to go down. Good. Yeah. Anything else? How else might disturbance influence diversity?
The bison likes to plant with grasses. So like if they keep on like continuing to prefer one grass and that, the other grass might have more of it.
Good. So you're going to need this picture. Good. Yeah, so you could increase evenness if disturbances preferentially remove the common species. If disturbances remove the common species for the most part, then the other rare species get more opportunity to grow and increase their population size. Good. Anything else? When we were thinking about succession, we talked about how over successional time, the total plant biomass goes up. Because first you have annual plants, then shrubs, then trees. You're not losing necessarily the annual plants and the shrubs along the way. You're adding the trees or the later successional plants through time. What was I going to ask there? So if you have a disturbance event and it wipes everything out, what's that early successional community going to look like? Do you think an early successional community is going to be highly diverse or dominated by one species and have low richness? Right. If you remove everybody, you know, it's Mount St. Helens and it's an ashfall site, right? The first species that get there are going to use all those resources. And then as later ones arrive and compete with them, then diversity starts to increase. But initially, within the disturbed patch, you're going to have low evenness and high dominance of one or a few species. So that's why I say it's complicated. And there's three different correct answers we've talked about just in the space of the last couple minutes. How do you think species, how do you think competition, similarly to the bison grazing that we just heard about, how might competition among species affect richness and evenness? If there's a lot of competition for resources among species, do you expect there to be one abundant species and a lot of rare ones, or lots of intermediate abundances all interactive? Your mumbles. If interspecific competition, the competition that occurs between species, is relatively weak compared to intrusted competition, what did we see in those lockable Terra models? We saw coexistence, right? That was the plot where everyone converges and both species are below their own carrying capacity, but they're both present. And so if you've got the right kind of competition relative to intrusted competition, then you might expect lots of species if they can all compete over time. If one species wins, then of course you end up with an uneven community dominated by one thing. All right, we've got about 10 minutes. I'm probably going to take a little bit of time next time. So we've got there's three indices or metrics of diversity that are used in the VNet all paper. One of them is called the Shannon's H. One of them is called Simpson's Diversity Index, or sometimes it's the Dominance Index. And then there's Piliu's J, which is a measure of evenness. I want you guys to think about how these two are going to be related to each other. As Simpson's Dominance Index goes up, what do you think is going to happen to the Evenness Index? These equations are all in the methods section of the paper, so you'll see them again. Shannon's Diversity Index, named after Claude Shannon, who developed it. And it's basically, it measures how confident you are in your ability to predict the identity of an individual species drawn from the community. So you imagine that your community is a bag of marbles, lots of different kinds of marbles, and you're reaching in there and you want to predict which color marble you're going to pull out. That's what this does. And it requires knowing the relative abundance of those different species. Or in the case of the marble example, if there's mostly red marbles and very few blue and green, then you can predict with high confidence you're going to pull out a red marble. But if it's even, you've got 30 reds, 30 greens, and 30 blues, you're going to have a 1 in 3 chance of being right. So that's kind of the analogy for how this works. And you just take the proportion of each species times the natural log of its proportion. So you do this product, and then you sum all those together. You're going to get a negative number because we're doing natural logs. So we make it positive again by making negative there. And it assumes that you're sampling individuals randomly, which is why I make the marble analogy, because you're just reaching into the bag. So think about how you do this. Break it out into the steps. Piliu's J is the metric of evenness that they use in VNet all, named for Evelyn Piliu. And one of the ways you can do that is just by taking Shannon's H and dividing it by the natural log of the number of species in that community, and that gives you this J. Sometimes you'll see people calculate it a different way, where you take Shannon's H and you divide it by what H would be if the community was perfectly even, which depends on how many individuals there are. But this is the one they use in the VNet all paper. And it says, well, basically, what would H be if all species were equally abundant in a perfectly even community? So it's affected by richness, and that's in the denominator for that, the richness. And the last one is Simpson's Index, which is more a measure of how much a community is dominated by one or a few species than it is a measure of diversity. And we use the statistic D, and you just take the proportion or the number of individuals of each species, multiply it by that number minus 1, and you divide that by the total number of species in the community times 1 minus that. So you get that ratio over and over and over again, sum it all up, and you get D. When people use this as a measure of diversity, they'll often do 1 minus D because indices are always bound between 0 and 1. That's what makes it a diversity index, between 0 and 1. And basically it's influenced strongly by how common the most common species are. This one has an advantage where if there's a lot of rare species or a very small number of rare species, you're going to get a similar number. What matters is how much of a drop there is from that first species to the second and the third. And how long that tail is doesn't matter as much. I've included this slide so you guys can think about how these things are calculated and how you might do that in a table. So here's our number of individuals in that community 2. Figure out how many there are total. Divide each of those numbers by the total. That gives you your proportions of each species. To calculate Simpsons Index, you'd also need to get the natural log of that number, which is right here. If you add all those up, you'll get, and you multiply them by themselves, then you'll get that H index. You can also do the same thing here, where you've got the 12 times 11 gives you 132. So you can do that for all of them. And then all of that is divided by 12 times 11. And that gives you that number. 132 divided by 12 times 11 gives you 0.0097. Notice these numbers are all quite small. You add them up and you get a number that's between 0 and 1. That's your dominance, your Simpsons D index.
How do you get the last one again? The last one?
Oh, sorry. Capital N is 117.
Oh.
Sorry. So the capital N is the total number of individuals, not the total number of species.
Okay.
Yeah. Thank you. That's why those numbers are so small, right? Because what's 117 times 116? Yeah, it's a lot. So now this number makes a lot more sense. Okay, and then we add all those up. And we can see what happens here. So this is our community 1, where our H index is 1.06, and our D is 0.58. If we go back to the other community, our D is 0.8. So this has a diversity. Oh yeah, this is our highly even community. So the D value is very low. And then this one is our very uneven community. So the D is much higher, 0.58. Okay, so we're going to talk about this last part at the beginning of class next time. It's 1:33 now. But I just want to leave you with this image that you've already seen once before. Remember, these are the watersheds. Within those watersheds, they have to know how many species are present, and they monitor that regularly. They have these transects set up. Each transect is a series of plots. Each plot is 10 square meters. So they go out there and count species regularly, so they know what the species pool is. And then in those little samples where we're going to measure for the Wien et al. Study, they're going to get some subset of all those species in each sample. So we're going to think about how species are going to accumulate along each transect at the start of class on Monday. Yeah, so we'll do that with the bison on Monday. You can leave your preparation homeworks right up here. I'll collect those, and I will see you guys then. I've got, so those of you who put it under my door, I have yours. But if you turn it in on Moodle, I don't necessarily have it yet.
I brought a stapler, but I put my name on both sheets.
Excellent. Well done.
I have the photosynthesis preparation due Monday.
Oh, yes. Sorry. Photosynthesis, after about 5 minutes on here or 10 minutes, we're going to start with photosynthesis. So that's going to be due Monday.
Does it have it?
Yeah, you can put it in. Yeah. Where's my mouse?